Hey guys, so in this video, we are going to go over some more histology. This is going to be the digestive system. So the GI tract, everything from top to bottom running through your body, as well as the accessory organs that are attached to the alimentary canal. So we're going to go through everything that we have as far as histology required on our lab manual. So first up, is going to be the tongue. The tongue is covered with a whole bunch of things called papillae, and papillae is plural, single is papilla. So there are these projections. Here's one right here. This is a singular papilla. And so we have four different types of papilla. They are going to be the circumvallates. The circumvallates are kind of pencil eraser shaped, relatively large circles on the back of the tongue. On the sides of the tongue, we have the foliate papillae. The foliate papillae look like pages of the book, and they look like book pages. That's why they have that name, foliate for folia for book. Other types of papillae on the top of the tongue are going to be the fungiform and the filiform. Filiform are thread-like, fungiform are mushroom-like, so they have circles too, but their circles are going to be much smaller than the circumvallates, and they cover the top of the tongue. So 3 of the 4 of these foliates, circumvallates, and fungiforms all have taste buds. So let's check out the features on this slide and see what we need to know and where we can find these things. So here's our single papilla, and our mouth is going to be subject to abrasion, abrasive foods, hot foods, drinks, and so we need to protect it. So our epithelial lining on the tongue is this stuff, and this is stratified squamous. So lots of layers of squamous cells, stratified squamous epithelia, stratified squamous epithelia on the top of each of the taste buds, on the top of the papillae. And so that's going to protect us there. But other special features that we need to know are those taste buds that we just talked about. 3 of the 4 of the papillae have them. Filiform does not have them. But here are the taste buds. So if you look, these little guys right here, these are all taste buds on the edge of this papilla on either side. So here's one, here's another one, here's some more. These are all taste buds. So this is how we taste our salty foods, sweet foods, all that good stuff. Those are the receptors. So you can see those on the slide for the tongue are the taste buds. I personally think it kind of looks like a toothbrush. Like here's the handle of the brush, right? And these are taste buds or all the bristles of the brush. So that is what we need to know on the tongue. Recognize that you are looking at an individual papillae or an individual papilla or multiple papillae. What is on top of the papillae that we got stratified squamous epithelial, and that underneath those down on the sides are going to be the taste buds. Next up is going to be the tongue. After the tongue is going to be the stomach. On the stomach, we need to know layers. I always like to talk about layers first on the stomach. There's quite a bit to know, but let's just start with the 4 layers. We have the mucosal layer, and that is going to be first, touching the lumen. So lumen is just the hollow, empty area where food will be passing through. Any of our hollow organs, our respiratory passages where the air goes through, the food goes through, and the GI tract, your blood flows through the blood vessels. They all have an open area called a lumen. So here's the lumen here, and then this is going to be the top. So the tops of anything exposed, we talked about in AP1 is always epi. So here is our mucosal layer, and it has the epi, and the epi is simple columnar epithelial for the rest of the GI tract. So at the ends where possible abrasion may happen, stratified squamous, but for the rest of the GI tract, it's going to be simple columnars. So this is our mucosal layer. It starts at the lumen and it runs to this kind of light peak band of smooth muscle right there. This kind of clear area, I think it looks a little clear, is right here. This is the submucosal area, and it looks a little clear on the slides, but it is going to be filled with the blood vessels, the capillary beds, and lymphatic drainage, lymphatic capillaries that are going to absorb in our food and our nutrients out of our stomach. So we are going to have the submucosal layer that runs underneath the mucosal layer for the stomach, the small intestine, all that good stuff. So we have mucosal so far. Submucosal is here. Then on the stomach specifically, all of the layers of the GI tract are going to have this area, muscularis externa, and it is a layer of smooth muscle. In the stomach in particular, kind of around there, it looks like, is where that smooth muscle stops. The stomach in particular, it has a big, thick layer of smooth muscle. For the rest of the GI tract, we're going to have some longitudinal muscle, and we're going to have some transverse muscle. But in the stomach, we also have a third layer of smooth muscle, and we have an oblique layer of smooth muscle. So that allows the stomach to really mix the food and churn it all together with the secretions. So there is a very chunky bit of smooth muscle here in the muscularis externa. And then last but not least is going to be the serosa right here. So this fourth layer is the serosa. So to summarize the layers, mucosal layer is here, submucosal was here, muscularis externa is all of this kind of hot pink smooth muscle layer, and the serosa is on the outside, the connective tissue that kind of holds the stomach in place is serosa. If we zoom in to the mucosal layer here on this middle slide, we can see it a little bit better. Definitely, again, simple columnar epi that line this. The stomach are going to have wrinkles called rugae, so we can kind of see one of those wrinkles kind of projecting up right now. On the GI tract, wrinkles equal more surface area. So we're going to have wrinkles in lots of different places, and depending on where the wrinkle is, it may have a different name. But in the stomach, they are called rugae in the stomach, and it just means wrinkles. Wrinkles. When we zoom in on the exposed mucosal layer, we have all of this simple columnar epi here, but if you look, we got lots of wrinkles coming up. So we're going to have villi. If we look on individual cells, we're going to have microvilli. If we go down, follow this, you can see it kind of dips in. I hope you can make out that it kind of dips in. It does it on this side too, kind of goes down and it dips in. Those are called gastric pits. Gastric pits. So the top are going to have the simple columnar epi up here, so all this simple columnar. But down here, here's a pit, here's another pit, here's another one. The gastric pits are going to be lined with different cells, chief cells, parietal cells, enteroendocrine cells that are going to have different secretions that we need to know in lecture. But you don't need to be able to identify the different cells in the gastric pit in lab at this time. You just need to be able to recognize that there is a gastric pit. On our mucosal membrane, we said that the epi is the simple columnar epithelial. There is also goblet cells in here because the GI tract is a big mucous membrane, and the mucus is produced by goblet cells. We mentioned those guys back in AP1. I don't really see one in this picture. I know they're there. Ooh, that might be one. They kind of show up clear sometimes with the stain. So that might be a goblet cell. Goblet cell over there. But we'll have goblet cells that produce the mucus that line all of our mucosal membranes. So that should cover everything that we need to know on this histology for the stomach. Moving on to an accessory structure. So we've got the liver. The liver is going to have functional units called lobules. So here's a lobule. Here, no, go back. Here's a lobule. And so those are going to be our functional units. The lobules are going to be separated by little walls. Little walls are called septums. And since they're in between the lobules, they are interlobular, because that makes sense. Interlobular septums are the little walls in between the lobules. Every lobule is going to have a central vein, these little white, clear hole looking things on our slides. Those are the central veins right smack in the middle of the lobule. The lobules are going to have little walls, the septums, to separate them, and occasionally you're going to see these little triads. There's another portal triad, and the portal triad is going to have three things in it. It's going to have bile ducts, it's going to have hepatic portal vein, and it's going to have the hepatic arteries in there for those little portal triads. If we zoom in on a lobule, so here we are, we've zoomed in this whole thing, it would extend out, and we probably have this bit, all of this is probably one big lobule, and we've zoomed in. On the lobule, we are going to see the individual cells now. So we've zoomed in and we can make out individual cells. Anytime you hear hepatic, hepatitis, hepatic portal vein, we are talking about liver. So the cells of the liver are called hepatocytes, because we know cytes mean cells. So hepatocytes are the cells of the liver, and you can see all these individual hepatocytes, all these individual cells. And one of the many, many jobs of the liver is to make a substance called bile. It's going to make it out of recycled red blood cells. It makes this bile, and it's going to produce it by the hepatocytes that secrete it. And then we can move that stuff down and store it in the gallbladder. So one of those jobs is to make the bile, and the hepatocytes do that. We also are going to have the little areas here. Maybe you can make out these little clear sections, these little sinusoids. This offers a pathway for us to connect the central vein right smack in the middle, and we can see it kind of sneaks on through till eventually make it to another one of these portal triads that just kind of allows the things to flow between the two areas, between the portal triad, make its way through the lobule, and we got our central vein going through these little openings, these little tiny thin areas called sinusoids coming through here. Next up, gallbladder. So we said the liver is going to make the bile, the gallbladder is going to store the bile. So anytime you eat any sort of fatty foods in it, we need to break down that fat with some lipase, but lipase is going to work better when you emulsify it and break the fat from big blobs into smaller blobs. That's called emulsification, and bile does that for us. It's like a Dawn dish soap. It emulsifies the fat into smaller blobs, and then our lipase helps break it down. So the bile is going to do that for us, and that is stored in the gallbladder. So on this slide, I zoomed out, I'm on scanning power, and this is like the edge of the gallbladder right here. So there's its serosa layer right there. It's muscularis externa, smooth muscle layer is going to be here, this kind of hot pink stained stuff. Is submucosal is really, really thin. It's kind of hard to make out. There's a little area right there. Could be submucosal. And then the stuff for mucosal is going to be simple columnar, like we mentioned for all the other stuff. We've got simple columnar, all this stuff at the just inside of my pen marks because I want to doodle over top of all of it. But all of that is simple columnar that is lining the inside of the gallbladder. And the gallbladder also has wrinkles. The wrinkles in the gallbladder are also called rugae. Rugae. Wrinkles in the stomach, rugae. Wrinkles in the gallbladder, rugae. Next up, though, are the small intestines. And with the small intestines, those wrinkles have a different name. So those wrinkles are called plicae circularis. That's the name of their wrinkles. And again, we said the more wrinkles, the more surface area, the better surface area, the more absorption you were having. And that's the whole goal of the small intestine, is to absorb your nutrients in the small intestine. So we have the plicae circularis, the big wrinkles. That leads to villi, the smaller wrinkles. And then if we go down to the individual cells, even the individual cells have wrinkles. And those are called microvilli. So we said that the cells that are going to line the tops of all of these areas of our small intestines, those guys are going to be simple columnars. Here we are, simple columnars. So they're going to be column shaped. We got an apical surface up here, a basilar surface down here, and at the top of their apical surface, this is the microvilli that gives them these kind of like finger-like extensions. I kind of think it looks like a hair comb, but that's the microvilli on an individual cell. So none of these pictures are really, really zoomed in that close that you could see it, but just know that it's there. These are all the tops that are covered in the simple columnar epithelial lining these areas. If we talk about the different areas, do know the different areas for both lecture and lab. So coming immediately off the stomach, we are going to go into a portion of the small intestine called the duodenum. Duodenum is pretty short. I think it's about 10 or 12 inches. It's not too long. But it is going to have lots of wrinkles, intestinal villi, like we were just talking about. It also has other special features that you might need to know in the histology. Are these kind of lighter area, kind of like bubbles, sort of kind of this stuff down here? Those are Brunner's glands. We find them at the base of the duodenum cells, at the bottom of that membrane layer. Moving on down from the duodenum, we're going to go into the middle area, the jejunum. The jejunum is much, much longer, around 6 feet. It's going to be a lot bigger than the duodenum was. It is going to have, again, the same epi, the simple columnar epi. This is kind of a decent shot to see some goblet cells. They are the clear cells. At the tip of the pointer of the arrow are the goblet cells. And what else do we have on this one? On the jejunum, we have the crypts. So if you follow all of these down and underneath that mucosal layer down here, all of these things are the crypts. And then last but not least, the last stop of the small intestine before it heads out of the small intestine, goes to the large intestine, is going to be the ileum. And so the ileum, here's its mucosal, submucosal, and we are going to have this area, all these purple guys, these big purple guys are called Peyer's patches. And the Peyer's patches are going to help with our immune system. So this tissue is going to house some of our lymphocytes and help us fight off things that don't belong in our body. They're going to be helping us out with our immune system. So these are the three different parts of the small intestine. Duodenum starts at the top, then we have jejunum in the middle, and we have ileum, which is also several feet long, in the very, very last stop before we head out of the small intestine and go into the large intestine. You can see on this little strip right here, before there's a tiny, tiny thin bit of serosa. But the rest of this stuff, the rest of this hot pink stuff, that's going to be muscularis externa. So that's going to be the smooth muscle. This light pink strip right there is the smooth muscle on the muscularis externa as it moves the food from point A down the tract to point B. Last but not least is another accessory gland. It's going to be our pancreas. Pancreas is super cool because it has both endocrine, and that would be the light pink stuff on the slide. Endocrine are the islets of Langerhans. The islet of Langerhans are pancreatic islets. They are the light pink stuff. All of this light pink tissue is all islet of Langerhans. This is islet of Langerhans, and this is the endocrine stuff. Not really what we're focused on in this chapter. Here is another islet of Langerhans, another one. Here is one zoomed in. This is pancreatic islet, or the islet of Langerhans. We don't worry about that in this chapter. For digestion, we want to know about its exocrine features. So the exocrine features are going to be the acini. So all of these purple cells, all this stuff, all the dark purple colored stuff is the acini acinar cells. So don't look at this. It's not this. It's all the rest of this, all of this purple stuff. This is all acini. And the acini's job in the pancreas is to make all of our exocrine secretions for our GI. So we talk about all the different macros that we break down in our GI tract. We break down carbs with amylase. We break down proteins with proteases. We break down lipids with lipase. All of those things are being produced in the pancreas by the acini cells. So all of these beautiful purple cells, all in here, all of this purple stuff is acini. This is what we're looking at, not this for this chapter. So that is the pancreas. Be able to identify the acini cells. They are dark purple. The islet of Langerhans is the endocrine stuff, and it is light pink on the pancreas side. I hope this helps, and I hope you have a wonderful day.